Dear students, now we are going to discuss a transcription unit and gene. Out of transcription unit, we completed what is promoter and what is terminator. In between these two, structural gene is present. So now we have to discuss what is gene and what is a structural gene. Transcription unit and what is a gene? What is the definition of a gene? So here, definition of gene in terms of a sequence of nucleotides is not correct. What is sequence of nucleotides? ATGC, ATGC, ATGC. For example, a gene is represented by this type of sequence of nucleotides. Now the definition of gene is ATGC, 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 ATGC. So this type of definition for gene is wrong one, literally wrong one. The correct one is definition means that should be in descriptive form, in description only. So that's why the definition of gene, instead of a sequence of nucleotides, changed into a segment of DNA that codes for a protein. That is called as cistron actually. Cistron is a segment of DNA, some region of DNA that codes for a protein. Go back to out of two strands, how many are involved? One is involved. If one is involved, how much part is involved? For that one, the answer is a cistron. Not only cistron, promoter, structural gene, and terminator. Particularly, the cistron is very, very important. So, cistron definition in the form of descriptive form of definition. A segment of DNA with certain number of nucleotides codes for synthesis of a protein is called as a cistron. Okay, so now take a transcription unit. To understand the concept of gene, structural gene, we should take a transcription unit. So what is a transcription unit? Represented by promoter, structural gene, and terminator. So this is a one transcription unit with a here promoter, this one terminator. So already the description of a promoter, terminator completed. This one is terminator, this one is promoter. So simply I'm using P here. T here, promoter, terminator. Now the pending one is, what is a structural gene in between these two? This part is structural gene. Okay. So here you can take this one as a cistron. So cistron is segment of DNA codes for a protein. Okay. So in this structural gene, how many cistrons are present? On the base of number of cistrons present in this structural gene, we divided cistrons into two types. So there are two types of cistrons. What are they? So first one is polysystron. Polycystronic structural gene. Polycystronic structural gene. This one, monocistronic. Monocistronic structural gene. Okay, polycystronic, monocistronic. So here structural gene is represented by or with many cistrons or one cistron. Polycystronic structural gene is present in prokaryotes. This one eukaryotes. So present in prokaryotes. Present in eukaryotes. Okay, right? So how to understand the polycystronic structural gene? Here, take that part, structural gene part. Structural gene. Okay, so one of the strands, one of the two strands codes for transcription completed. mRNA produced. Here I'm taking mRNA, messenger RNA. Okay, so what is polycystronic structural gene? Many cistrons are present. So here, instead of a structural gene, but take a cistron 1, cistron 2, cistron 3. Many are present. Here I'm taking 3 cistrons. Cistron 1, 2, 3. So this is a tricistronic. Polycystronic means more than 1. 2, 3, 4, 5. Many are present. Here I'm taking 3 cistrons. You may take more than that also. So this one, 5, 3. So this is cistron 1, 2, 3. Take a cistrons as it is. Cistron 1, cistron 2, cistron 3. So this is definitely tricistronic, polycystronic structural gene, particularly tricistronic. Now, attachment of ribosome. 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 How many ribosomes? 1, 2, 3, equal to number of cistrons. How many cistrons here? 1, 2, 3. And what are these are? Ribosomes, 70s ribosomes, only 70s. Go back to first year Textbook definition, polysome. What is polysome? Attachment of a number of ribosomes to one mRNA is called as polysome or not? Polysome or polyribosome. So 70S ribosome, 70S ribosome, 70S ribosome, one kind only. And their number is equal to number of cistrons present on mRNA. So all these are 70S ribosomes. 70S ribosomes. And now this is called as polysome. Polysome or polyribosome. Okay. So now translation. What is cistron? A segment of DNA, codes for protein. So after translation, from this one protein, whatever the type of protein 1, and from this one protein 2, from this one protein 3, 1, 2, 3 proteins. So number of proteins produced out of this polysome is equal to number of cistrons or number of ribosomes. So that is about a polycystronic structural gene. Okay, right? Now, what is a monocistronic structural gene? Monocistronic structural gene is a little bit complicated, confusing one, but with the help of this flowchart, you can understand what is a monocistronic structural gene. Now, take a structural gene. This is a structural gene of a transcription unit of eukaryotes. 3, 5, 5, 3. Here, what about the number of cistrons? C1 I am taking, cistron is only one cistron only. Here I am writing C1, cistron 1. Okay, so this cistron is not continuous, discontinuous. What is that? A discontinuous cistron. So this one is a exon coding part, intron non-coding part, exon coding part, intron non-coding part. Okay, exon coding part. 1, 2, 3. Take one more also. Intron, exon. Same continue. Exon, intron, exon, intron, exon, intron, exon. So what is exon coding part? Intron non-coding part, intervening sequence. Coding part, non-coding part, coding part, non-coding part, coding part, non-coding part, coding part. Okay, right? Is it clear? No. So this cistron is divided into how many zones? Coding zone 1, coding zone 2, 3, 4. So 25% of that cistron, 25% of that cistron, 25% of that cistron, 25% of that cistron. So all these are 25%, 25%, 25%, 25% belongs to one cistron only. So here that 25%, 25%, 25% plus is equal to 100%. If all these 25, 25, 25, 25 are continuous, we'll get a complete cistron. But here non-coding regions are present. Simply signal available, no signal. Signal available, no signal. Signal available, no signal, like that. Okay, right? So that cistron coding part was cleaved into certain number of coding regions which are interrupted by non-coding regions. They do not code for protein formation. Okay, remember this one. So here, this cistron is cleaved into a number of segments or not. So this gene, the structural gene, this structural gene is called as a split gene. Split gene. So this is the way to understand the textbook content. We have to customize a textbook according to neat requirement. We have to tailor the subject at the time of learning, tailor the content according to competition. Otherwise, you can't understand. See, in your textbook given a split, what is that split? Cistron was cleaved into, divided into a number of parts. So simply divide this cistron into coding part into 25%, 25%, 25%, 25%. So that is a split gene. The structural gene present in eukaryotes is monocistronic one, and that is split gene, discontinuous. Now go back to the definition of cistron. What is cistron? A segment of DNA codes for protein, whatever the number of proteins. Here it is clearly that definition is applicable. Reason, this is cistron 1, 2, 3, continuous, continuous, continuous. There is no split gene. There is no intervening sequences, completely coding part, completely coding part. So segment of DNA coding for protein. Yes, definition is correct. Here, see, what is segment of DNA coding for protein? This is a segment of DNA. Is it coding for protein? No. Reason, introns are present. So the definition is complicated or not. If it is continuous, now we can say a segment of DNA codes for protein, acceptable. But here, coding part, non-coding part, coding part, non-coding part, coding part, non-coding part. How to define that one in terms of a segment of DNA codes for a protein? Complicated. The definition was complicated or not? Yeah, definitely. Reason, intervening sequences are present. So this is the segment of that cistron present in the structural gene of a monocistronic structural gene present in eukaryotes. So from this one, transcription completed, for example. This one is taking place within the nucleus, eukaryotic nucleus. Now, RNA is produced. What is that RNA is important? RNA 5, 3. Always polymerization of any strand, that is either DNA or RNA, whatever it is, DNA and RNA is 5 to 3 direction from a template with 3 to 5. Remember that one. So this is a heterogeneous. This one is a heterogeneous nuclear coming from the nucleus. Heterogeneous nuclear RNA. So simply this is called as a hnRNA. hnRNA. Heterogeneous nuclear RNA. Heterogeneous. Reason, exon, intron, exon, intron. Hetero or not? Different. So here also take them exon. Simply I'm taking exon, intron. This part is very complicated. With the help of this flowchart only, you can command the textbook content. Exon, exon. How many? 1, 2, 3, 4. Take whatever the number. Just for example, only I took that much number of segments. You can take more also. Those are the coding regions. Now, non-coding region. non-coding region, non-coding regions. 1, 2, 3, 4. 1, 2, 3, 4. Exon, intron, exon, intron, exon, intron, exon, intron. Okay, stop here, no problem. Okay, so this is a primary transcript. This one is a primary transcript. This one is a primary script. Primary transcript. Primary transcript. So this is a HNRNA, heterogeneous nuclear RNA. What about cistron? Divide into a number of coding regions, 25%, 25%, 25%, 20%. Intervening sequences. So intervening sequences must be eliminated. Okay. So now take splicing. Splicing means cutting, elimination of introns. Cutting. Exon part, intron part. Exon part, intron part. And continue it. Exon, 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 exon part. So exon, exon, coding part, coding part. And what about this one? Intron, 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 non-coding part. Cut. After cutting, only exon, 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 exon. All introns removed. Intron, 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 certain number of introns. All these introns removed. Splicing is regulated by RNA only. So this is a exon, 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 exon. Now ligation, attachment. Here enzyme required is a DNA ligase, DNA ligase. Attachment of all these 25%, 25%, 25%, 25%, 100%. Okay, after joining, we'll get a complete cistron. This is cistron now. Complete continuous cistron. 5, 3. So now this is called as mRNA. Some other processings required. We'll see that one later, not now. Up to exon intron concept enough. What about UTR regions, tail part, cap part? We will see that one in the next topic only, not now. Okay, right? Wait. So now this is called as a well processed mRNA. So heterogeneous nuclear RNA is subjected to resizing, tailoring, reprocessing. Primary transcript is not suitable for translation. Reason introns are present. So that's why introns eliminated. So in the final script, there is no intron, no non-coding regions are absent. In the primary transcript, non-coding regions are present. So in the final script, which is suitable for translation, introns are absent. Only exons are present. So exon 1, 2, 3, 4, 1, 2, 3, 4. 25%, 25%, 20 plus it, make it plus, addition, addition. 25 plus, 25 plus, 25 plus, 25 plus, 100%. So one cistron, complete or not? No. So now that is 100% cistron, 100% processed. So coding regions are, now coding part is continuous or not? No. Only one ribosome is attached. Only one ribosome. Only one ribosome. That is ATS ribosome only. This is not a polysome, monosome. ATS ribosome. Next translation. Translation and protein. How many proteins? One protein only. One cistron, one protein. So that is about a monocistronic structural gene. Without this flow chart, definitely you can't understand the content. Please follow this one. Customize the textbook content. Tailor the textbook content to the academic needs. Otherwise, you can't understand. You have to buy it. Okay, so just if you practice this one, you can teach this one to everybody. Okay, is it clear? Now, the conclusion. See, out of prokaryote and eukaryote, for example, here I'm taking one structural gene, here one structural gene. Which one is more efficient regarding production of proteins? This one. See, 1, 2, 3, many proteins equal to cistrons. Here, our mRNA coming from nucleus is able to produce only one protein. So biomass production is very less. Biomass production is more here, more efficient. One mRNA carrying information for many proteins. Here, one mRNA carrying information for one protein only. Is it clear? So that is about gene. So far we completed transcription unit completed, promoter completed, terminator, now structural gene. Okay, that structural gene is either polycistronic type or monocistronic type. Okay, my dear students. So that is about the complicated area of that transcription unit, the transcription part. From here onwards, complicated areas are present. Okay, so to understand those areas, please follow these flow charts. Okay, otherwise you will be at the crossroad. You can't understand, leads to confusion. Okay, bye, see you. We will meet in the next video.